The cellular level of organization. In our study, there are three fundamentally different cell types that exist. Prokaryotic cells lack membrane-enclosed structures. If you think of pro as before, as like a prologue. Eukaryotic cells possess membrane-enclosed structure. U means true. So this has a true nucleus that we're going to see as the main difference between eukaryotic and prokaryotic cells. For now, we're going to focus on eukaryotic cells, but be aware there's a third cell type. Archaean cells possess qualities of both prokaryotes and eukaryotes. They have their own domain. Starting with eukaryotic cells, this is the type of cells that our bodies are composed of and other animals. Eukaryotic cells are structurally complex. They have a true nucleus. They possess membrane-bound organelles. And it makes up animals, plants, fungi, and protists. This is the domain eukarya. Even though there's not exactly a typical animal cell, you'll see this typical diagram when depicting animal cells. It highlights many of the organelles that you will see in eukaryotic cells. So this is a typical animal cell here compared to a typical plant cell. We'll refer back to these diagrams as we go along. What I want you to notice in both of them, though, is that the main organelle, the largest one, and organelles, you think of it as a tiny organ. So we have this large purple sphere here in either the animal cell or the plant cell. Here I know we have this larger vacuole. We'll get to that later, but for now, we're going to start with the nucleus. This is the control center of the cell. The nucleus, again, is going to be that main organelle that you'll see under a light microscope. If we're looking at either plant cells or animal cells, these are cheek cells, this larger dot that you see within these cells, that's the nucleus. And so it's generally the largest organelle that you'll be able to see. It's about 5 micrometers in diameter. That's 5 millionths of a meter. The nucleus stores genetic material as DNA. DNA, we saw earlier in chapter 2, is deoxyribonucleic acid, and this is where genetic information is coded. We'll get into this a lot more later on. Every cell in an individual contains the same DNA, and DNA governs the characteristics and metabolism of the cell. This is why we call the nucleus the control center. Outside the nucleus is this double-layered envelope that has pores. The nuclear envelope separates the nucleus from the cytoplasm, and it's made up of phospholipids. Those pores are nuclear pores, and they're openings that permit transport of protein and ribosomal subunits. So it's going to let stuff in and out. Within the nucleus, we see the nucleolus that looks like a little nucleus. It's made of RNA and protein, and this is where ribosomal RNA is made and produces ribosomes. We also see chromatin. Chromatin is loosely coiled fibers made of DNA and protein. They shorten and thicken during cell division and then are called chromosomes or color bodies. These are the most important structures in the nucleus because they control the functions of the cell. This is the encoded genetic material. Looking within the nucleus, we can see this double membrane here, and it's made of a phospholipid bilayer, very similar to the plasma membrane of the cell. The nucleolus looks like a nucleus within the nucleus. These fibers here are chromatin, and this is DNA and the proteins that it coils around. And here we have the nuclear pores that are going to allow things in and out of. All cells are surrounded by a plasma membrane. This forms the boundary that separates the contents of the cell from the surrounding environment. So in a lot of ways, the plasma membrane is what separates living from non-living, particularly if you have a unicellular organism composed of one cell. The plasma membrane is going to regulate the entrance and exit of molecules into and out of the cytoplasm. This controls what comes in and out of the cell, basically. It is semi-permeable, meaning that some things can freely enter and some can freely exit, and others cannot. Some things can easily permeate that membrane. It's made mostly of proteins and lipids. We'll look at these in the next two figures. This is a section of the plasma membrane. So if we're looking back at a cell, so the plasma membrane is what surrounds it. And we've got a couple different sections here that we're going to look at that give a little more detail. So cross section here, the plasma membrane is also referred to as a fluid mosaic. If you think of a mosaic that has a lot of little pieces that make up the whole, the plasma membrane is similar. But here, it's a fluid mosaic because these pieces move and they're not static. So what we first want to look at here is primarily we have a lot of these kind of blue spheres with little tails hanging off of them. Those are phospholipids. We saw these back in chapter 2, and they form this phospholipid bilayer. But within that, we also have proteins and we have cholesterol. Figure 4.1 gives us more detail on this phospholipid bilayer and the fluid mosaic that is the plasma membrane. So if we're starting to look at pieces of the plasma membrane, the structure and function there, we have lipids, two types. We have the phospholipid bilayer. Within the phospholipid bilayer, and looking at the chemical properties of that, we have hydrophilic or water-loving polar heads. They face inside and outside the cell where there's water present. And those tails are nonpolar or hydrophobic. They're water-fearing, and they face each other away from the water. Let's look at that again. So we have these hydrophilic heads, hydrophobic tails. And so the hydrophobic tails that are nonpolar face one another, and hydrophilic, water-loving, heads that face outside and inside the cell where there's more water. We also have cholesterol in animal cells that controls excess fluidity, gives it some structure, and adds strength. The cholesterol, remember that cholesterol is also a lipid, is also hydrophobic. So again, those cholesterol molecules are shown here. And being hydrophobic, they're going to face the interior of that phospholipid bilayer. The amount of cholesterol within our plasma membrane varies throughout the year. So if you're someone that gets your cholesterol checked, it's good to have it checked twice a year, like in the summer and the winter, since these levels change. So as we were looking at these phospholipids, if you're looking more at the chemical nature and the polar head and the fatty acid chains that form the nonpolar tails, they spontaneously form this bilayer in which the hydrophilic heads face outward towards watery solutions and the tails form the hydrophobic interior. When we're looking at the permeability of the plasma membrane, this is important. The plasma membrane, it's not like plastic wrap where things aren't going to move in and out very easily. Instead, it's selectively or semi-permeable. And so in this way, the plasma membrane can regulate the passage of molecules into and out of the cell. Which molecules can freely cross the membrane and which may require proteins and or energy depends on the size and the nature of the molecule, that being its polarity or its charge. Amino acids, which make up proteins, right? Proteins themselves, sugars, which are a type of carbohydrate, and water cannot freely cross the membrane. They require special ways to get through. However, small, uncharged molecules freely cross the membrane. For example, carbon dioxide, oxygen, glycerol, and alcohol are all oil-soluble. If something is oil-soluble, then it can freely pass through the interior hydrophobic region of the nonpolar tails. They slip in between those hydrophilic heads and pass through. Another piece of the fluid mosaic are embedded proteins. We're going to look at two types here. We have channel proteins that provide a passageway here, as well as carrier proteins that they provide a passageway for molecules, but they combine with the substance to help it move across the membrane. With a channel protein, as you see, it's providing a channel, right? It allows a particular molecule or ion to cross the plasma membrane freely. Cystic fibrosis, an inherited disorder, is caused by a faulty chloride channel. A thick mucus collects in airways and pancreatic and liver ducts, so that this channel, if we were doing an example, that allows the chloride ion to pass whenever it's faulty, then you have an excess that builds up. Carrier proteins selectively interact with specific molecules or ions so that they can cross the plasma membrane. An example is this family of GLUT carriers that transfers glucose in and out of various cell types of the body. So glucose is a type of sugar. Different carriers respond differently to blood levels of glucose. Some eukaryotic cells have cell walls as well as an outer border. Plant cells may have a primary and secondary cell wall. Cellulose is the primary component, and this adds strength. Lignin is found in the secondary cell walls for even more strength. If we're looking at that, back from chapter 2, looking at the composition of cellulose, we mentioned that it's made of glucose units, but that the structure, they're joined in such a way as to give strength, rather than being a storage form of glucose, as we saw with glycogen and starch.